Good afternoon, everyone, and thank you very much for your kind introduction. And yeah, sorry about this swap this morning because of the flow of the talks and everything. So it would be nice that Dr. Ralad would present, his work in the morning instead of me. So today I'm going to talk about my latest and recent work on identifying molecular targets of therapeutic agents using combined metabolomics and in silico docking. So as you may know already about cholangiocarcinoma or bile duct cancer or CCA from Dr. J.Phetcharaburanin previously. So bile duct cancer is an epithelial malignancy of either intra or extrahepatic biliary tract, and this is considered as the second most common liver cancer following hepatocellular carcinoma or HCC that accounts for 10 to 15% of all hepatic cancers. And overall incidence, well, the global incidence shows on this map here, and to focus in Thailand. So as you can see, Thailand has the highest incidence of CCA in the world. In particular, the northeastern region where Khon city is located. So we are so-called the capital city of OV infection and also cholangiocarcinoma, which is unfortunate. And this slide shows the resected liver cancer tissues that you can see. Actually, there is the J.Phetcharaburanin vivarini or OV, which is a liver fluke that is a major cause of the cholangiocarcinoma in many countries in Southeast Asia and also in Great Mekong sub-region countries like Thailand, Laos, Burma, Cambodia, and the southern region of China as well. So they live in here after ingestion of the raw fish containing the liver fluke. The liver fluke can persist in the biliary tract for 20 to 30 years until that particular person gets these symptoms, and then they can be diagnosed at a very late stage, unfortunately. And this is the real liver flukes that the surgeons in our Sinakrine hospital took out from the patients. Well, it's just maybe Dr. Vasin here can tell me how many % of the total amount of the liver fluke taken out from the human biliary tract, but maybe just 10%, that's what I've heard. So far, the gold standard for the detection of the cholangiocarcinoma in Thailand is actually based on ultrasonography. So as you can see, the first three echo patterns look very normal. Oh, sorry. The first three panels of the ultrasonographic pictures here show the normal echo pattern, so very clear bile duct in comparison to the bottom trees. So as you can see here, there is the starry skies and also the cloudiness of the bile duct showing the dilatation of the bile duct. So far, the Cholangiocarcinoma Research Institute is still using the ultrasonography as the gold standard technique to detect cholangiocarcinoma, to detect OV infection that are more prone to have cholangiocarcinoma. It doesn't work for some reason. Okay, so and the chemotherapeutic drugs that our clinicians use nowadays are the cisplatin combined with gemcitabine, which is first-line treatment for CCA chemotherapy. However, this treatment seems to have or incorporated with the side effects, for example, diarrhea, hair loss, vomiting, shortness of breath, and heart failure. So scientists in the field are trying to focus more on the naturally derived agents, for example, medicinal plants that contain secondary metabolites, and they are proved to have more efficient and safer potency. So what are the secondary metabolites? So we need to jump back one step to this primary metabolites. Primary metabolites are those metabolites that are essential for growth, development, and reproduction, whereas secondary metabolites are those that have been produced on a specific purposes and they are considered as optional metabolites for advantages or certain advantages of the organisms. And there are so many applications of the secondary metabolites that we can do. And these metabolites, again, can come from different sources in nature. For example, plants, fungi, bacteria, and so on and so forth. And it can be used as the medicinal herbs, pills, some injections like Botox, and it also has some effects against cancers as well. And this slide shows the properties of the phytochemicals, including anti-proliferative, pro-apoptotic, anti-angiogenic, immunomodulatory, antioxidant, and anti-inflammatory properties. So in terms of mechanisms of the phytochemicals, it includes three major mechanisms. One is the modulation of the signal transduction pathways. Two is the modulation of gene expression involved in the mechanism of the molecular targets. And three is the interference of the metabolic processes through the disturbance of the enzymes. So having talked about metabolites, so I would just like to remind everyone about metabolites again. So metabolites is not part of the central dogma, but is actually the part of the end products from the central dogma. So as you can see from the flow of biological data map on this slide here, you can see that metabolites are actually the consequences of the metabolism after it takes place. And again, the secondary metabolites of phytochemicals are considered metabolites. We can use metabolomics to probe or to profile the chemical composition of the metabolites from the secondary naturally derived organisms. And having talked about metabolomics, there are actually two major approaches. One is targeted metabolomics, and the other is untargeted metabolomics. When we perform targeted metabolomics, is that we are going to do a targeted analysis on the known compounds of interest. So that means the identification is already known, and this is hypothesis testing strategies, and this is to validate the potential biomarkers, for example. And when we conduct the untargeted metabolomics, that means we do global profiling, and this is considered hypothesis generating strategies, and it needs the database to do some identification and metabolite assignment, and it can be used for biomarker discovery. Now, I would like to show you the comparison between the use or the application of metabolomics and in comparison to the conventional screening of the phytochemicals or bioactive compounds. So before, on the left-hand side, this is what we are very familiar, especially when we work in phytochemical research in Thailand. So we need to have the crude extract, and then we do the differential fractionations, and then we obtain different fractions from the fractionation experiment, and then we test each fraction against the cells, or we use each fraction to do some experiment for bioassay, right? Then this is going to take forever to finish until we could identify the compounds or the bioactive compounds that have the major activity against whatever we are going to study. So in our group, so we propose that we can use metabolic profiling based screening technique in order to profile the biochemical constituents in the crude extracts, and then we use the with the help of chemometrics, so we can identify the potential biomarkers that have the roles, biological roles towards the particular study that we are going to test. It could be either animal studies or in vitro studies. So the first study that we conducted using this approach is a plant-derived anti-cancer agent against cholangiocarcinoma that was conducted by Dr. Bandit Promlaksa, who is now working in the Department of Biomedical Science at the Ministry of Public Health. So at that time, he was our former PhD student who published a paper on evaluation of anti-cancer potential of Thai medicinal herb extract against cholangiocarcinoma cell lines. In this study, he used 3 different Thai herbs, including S. dausis, C. helicacabum, and G. psilocybin. Or in Thai, grot nam, kokaom, and ban meruroi pa. So I'm sorry for the jumping words due to the cross-platform. So I prepared my talk on Mac and then I transferred to the Windows. So, but in short, we would call plant A or grot nam in Thai as ESD, kokaom as ECH, and ban meruroi pa as EGC. Then we perform the phenolic content and antioxidant assessment to measure the phenolic contents and the antioxidant properties. And we found that grot nam or ESD shows the highest amount of the phenolic contents and also shows the highest antioxidant properties amongst the 3 plant extracts. And then we treated the two different cell lines in cholangiocarcinoma, KKU100 and KKU213. So these two cell lines show the different degrees of proliferation and aggressiveness of the cancer characteristics. So we treated the two cell lines with the ESG extract and also other extract at different time scales, 48 hours and 72 hours. And then we performed the colony forming assay to visualize the effects of the plant crude extract on the CCA cell lines. And then we found that GROTNAM or ESD extract shows the promising lower cell viability of the cholangiocarcinoma cell lines of the two types. Then we perform the correlation regression analysis to see the potential metabolites that are associated with the lower cell viability, and we found that lignin compound 2012 seems to be the promising metabolite that has such a function. And then we confirm the existence of the lignin compound 2012 using the 2D NMR, and COSI is one of the examples. And having talked about lignin, so lignin compound has three different domains or components, chemical components. One is cyanide alcohol, two is coniferyl alcohol, and three is p-coumaryl alcohol. So then we try to synthesize this lignin compound in our lab, but then it's quite difficult. We found it very difficult actually to join, the three parts of the chemical components together. So, and we also looked up on literature, and we found that after this lignin compound 2012 has been dissolved in, let's say, water, so it's going to give out, the three different chemical domains, as separated. So that's why we use, the three different chemical components from lignin compound 2012 to treat cholangiocarcinoma just to validate our findings. And we found that coniferyl alcohol seems to have the effect mostly among the other two. So at first, we found that metabolomics can surpass several steps of conventional method for identifying bioactive compounds in plant extracts, and also it can highlight the association between the metabolites and the phenotype that we observe. Then we perform another study using the synthesized coniferyl alcohol, or CA, against the cholangiocarcinoma. And as you can see here, so we treated our CA to cholangiocarcinoma cell lines, again KKU100 and KKU213, and we found that at the 48 hours and 72 hours, it shows the lower cell viability. And then we performed the flow cytometry assay, and we found that the early apoptotic and the late apoptotic rate of the cells are mostly dense. So that means the cells seem to be turned into apoptotic pathways after being treated with CA compound. Then we collected the cells and also the cell culture media. For the cells, for the cell pellets, we extracted the intracellular metabolites and run that on NMR and performed the statistical analysis. And then we found that for KKU100, the model is not valid actually. So if we look into, however, if we look into the KKU213, the model is valid, and we can see the discriminatory metabolites that are increased in concentration when the cell viability is low. And these include glutathione, succinate, and taurine, and also some of the phosphatidylcholine species. Then again, we also extract the metabolites from the extracellular components, which is the cell culture media, and we perform the statistical analysis to see the extracellular metabolite alteration after the treatment of the CA compound. And when we found that, again, KKU, but this time we can see that there is the metabolic alteration after being treated with CA compound in the KKU100 that can be reflected in the extracellular compartment, and also likewise to the KKU213. Then we could come up with a schematic diagram that can explain and help aiding the understanding of the apoptotic mechanisms of what CA exerts into the KKU cell lines of both cell lines, KKU100 and KKU213. So, and these are in accordance to the proteins that we have found, change in accordance to the CA compound treatment. And mostly it's about apoptosis, and also it enhances oxidative, it lowers oxidative stress and lower phosphatidylcholine compounds. Then, well, from this finding, we then, however, have some questions like how or what is the target of the CA compound, right? So we know that the CA compound exerts some effects on the CCA cell lines. It can kill the CCA cell lines through apoptotic pathways, but how? And what is the target? So we listed up the metabolites that have been significantly altered both inside the cell and outside the cells. And we've come up with this table here as a summary. And then we also correlated our findings to the apoptotic proteins involvement like BAX and BCL2. And we found the association between the increase of BAX that has been associated with a lower level of taurine, and also the downregulated BCL2 expression that is associated with the lower level of NAD+. So, however, the direct molecular targets interact with coniferol alcohol are awaiting to be elucidated. So, we come up with this plan. So it is the combined metabolomics and in silico docking approaches. When we start with global metabolomics that can apply to screen the dysregulated metabolites, right? And then we perform the pathway enrichment analysis and enzyme selection. After that, we conducted the in silico docking experiment. And then we could identify the existence of the protein targets, and some candidate targets will be detected. And finally, we can validate the targets that we perform from the in silico docking. So this study has been mainly conducted by my current MSc student, Sirina Pasulamnat, with the research aim is to identify the enzymatic protein targets of coniferol alcohol in the apoptotic pathway and its molecular mechanism on CCA cell lines. And we've got the hypothesis is that there will be specific enzymatic proteins related to such metabolic changes induced by coniferol alcohol in cholangiocarcinoma cell lines that can be further used for CCA therapy. So our research questions include what are the enrichment metabolic pathways affected by coniferol alcohol in CCA cell lines? What are the enzymatic proteins targets of CA compound in CCA cell lines from in silico docking study? And finally, what are the molecular mechanisms of the CA in anti-cancer activities against cholangiocarcinoma cells? So from Promraksa study, we ended up to this point where we found the dysregulated metabolites from his analysis. Then we selected these metabolites to perform the pathway enrichment analysis, and then we did the candidate enzymatic protein selection. We conducted in silico docking and also the potential novel target identification for CCA therapy. So let's start with the pathway enrichment analysis. So the significantly changed metabolites have been recruited into the model, and pathway enrichment analysis using cytoscape equipped with Metaboscape toolbox was performed. And then candidate protein list represented by enzyme classification or EC number was chosen. After that, we collected the EC numbers from the pathway enrichment analysis. Then we searched the gene names that have expression in the liver tissue by gene card, followed by finding information about representative protein structure linked to the PDBE, which is a protein data bank in Europe. Then we need to consider whether the PDBE does provide the desired protein structure. If yes, we then download the protein structure, and then we end this process. If not, we continue. So we need to determine further the UniProt from the EC number by using gene card. Then we make the protein homology modeling for UniProt using Swiss model. And then we consider again whether does templates have sequence identity higher than 40%. If yes, then we selected the template with highest coverage and resolution and download the protein structure and end this process. If not, we need to consider further whether templates have the sequence identity higher than 30%. So basically, we lower down our cutoff. If yes, we consider another time for the templates, whether they have coverage higher than 40%. And if yes, we select the template with highest coverage and resolution, download protein structure, and end the process. If not, we download the computed structure for UniProt from AlphaFold. So these are the optimized sort of algorithmic pipeline that we performed from the previously published article. So now let's come to the system preparation for docking. What we need is one, the protein structures from either PDB, Swiss model, or AlphaFold. Two is the CA, in this case, coniferous alcohol structure from the zinc database. Three is the all potential binding sites, or we call the pocket, grid box identified by F-pocket. So after we've got these three components, then now we are ready for molecular docking. Then we perform the molecular docking using the orthodox reader. I'm not going to go through all the equation and the vectorization of the equation. And yep. Then we perform the pathway enrichment analysis, right? We found these metabolites that are the key change in metabolites from the treatment of the CA compounds. And this is our second component, which is the enriched pathway-related metabolites of KKU100 and KKU213. As you can see, the treatment, the same treatment applied into different cholangiocarcinoma cell lines affects different metabolic pathways, right? And also, in turn, reflect different protein targets as well. And this could be depending on the different characteristics of each cell line, too. Then, after performing molecular docking on KKU100 surface proteins, we selected the top 7 proteins that have the lowest binding energy or score. And as you can see here, the top 2, which are spastin and also glycerol 3-phosphate dehydrogenase 1, have this binding score at -8.4. So the lowest binding energy suggests a strongest interaction between the ligand and the receptor, and that results in potential targets of coniferous alcohol, as we can observe from this experiment. And now let's have a look on the KKU213A. So we selected the top 6 on this KKU213A to show, and we found that the top selected one is the homology model for cytochrome B5 reductase 1, and it has the binding energy score at -9.5, and it could be also serve as a target for CA compound as well in KKU213A. Then, having had the top ranked protein as a candidate enzymatic proteins for KKU100, so we found that spastin is actually the AATP-dependent microtubule severing enzyme that has roles in various cellular processes, and GPD1 is actually the enzyme that catalyzes the conversion of the dihydroxyacetone phosphate, or DHAP, to glycerol 3-phosphate, and it also plays a key role in cellular metabolism such as glycolysis. And then another protein that we think it could serve as the target is the D3PD1. And as you can see here, this is the binding site of the CA to the D3PD1, and this would enhance the binding of the NAD+, that is a coenzyme for GPD1 to work. So that means if the CA compound that it added into the assay can bind to the binding site here, it can promote or enhances the binding of the GPD1 and NAD+, and that would even stimulate or trigger the effect of the enzyme and enhance the apoptotic pathway to be upregulated. And if we check on the gene expression profile across all tumor samples and pair normal tissues, we can see that the D3PD1 is actually highly expressed in normal cell, interestingly, not in the tumor cells. But that means there would be some certain characteristics that there is the GP3D1 binding to the ligands at some point, and that would promote apoptotic pathways. However, this is still under further investigation. And again, activation of the GPD1 can decrease NAD+ and DHAP levels that can satisfy with metabolic changes in KKU213, as can be evident by the previous study of Plomraksa et al. So the two findings, one in Metabolomics and the other one in our current study, which is the protein targets, go accordingly. And GPD1 also promotes the calcium influx and apoptosis of the tumor cells by the lysophosphatidylcholine or lyso-PC, platelet activating factor receptor, transient receptor, potential vanilloid 2 axis in the bladder cancer that has been proved already in other cancer. And also in breast cancer, scientists show GPD1 act as a tumor suppressor through increasing PI3K and AKT-mediated lipid metabolism signaling pathway as well. And for the top-ranked protein of candidate enzymatic proteins for KKU213A, we propose that it is a cytochrome B5 reductase 1 that have the function in lipid peroxidation and ferroptosis. Ferroptosis is actually a form of regulated cell death characterized by the iron-dependent accumulation of lipid peroxides to lethal levels. And this particular protein has the overexpression in CCA that contradict to the CYP5R1 have reported role in ferroptosis and colorectal cancer. And if you look at the binding site here that we simulated, this is a binding site of the ligand, in this case is CA compound that we synthesize binding to the cytochrome B5 reductase 1. And we show that the energy of binding is here. So in conclusions, at the moment of this study, we proposed the optimized pipeline to combine metabolomic findings with in silico docking that can be used to identify the molecular target of the novel bioactive compounds, especially in the crude extract of plants in this case. So what is the bridge between metabolomic and in silico docking? One of which is the pathway enrichment analysis. And the second step is the filtering by database. So that means database also plays role in fulfilling the requirement of selection criteria. And in silico docking is next, and last but not least is a validation. So we could validate our finding using either molecular dynamic simulation or we can do a wet lab work. So in this study in particular, we found that coniferous alcohol may target spastin, GPD1 in KKU100 and cytochrome B5 reductase 1 in KKU213A. Coniferous alcohol may also induce apoptosis in KKU213A by activation of GPD1. And finally, if coniferous alcohol could enhance the binding of drug to receptor on target cells, it may improve the efficiency of drug in co-treatment with the chemotherapeutic drugs. Last but not least, I would like to thank all of my collaborators and colleagues in Khon University, and also from Mahidol University, Prince of Songkhla University, Nanotech Thailand, Murdoch University, Imperial College London, and I would like to thank all the funders that provided the funds and budgets to certain studies that I've shown. And thank you to Royal Society of Chemistry, United Kingdom, eAsia, Kallaprapruek Foundation of United States, National Research Council of Thailand, or NRCT, Ministry of Higher Education, Higher Education, Science Research and Innovation, such a very long name for this ministry. And I would like to thank Khon University, Kallaprapruek Research Institute for providing the initial funds as well. Last but not least, thank you all for your attention.
Okay, because of the limit of time, we are around just only one question from our participants. Any questions?
Fascinating work with the look at the target of the metabolites. I think finding the metabolites itself is a very long process. Now next stage is to look at the target. Yeah. I'm not working in the field, find the target, but I do have a collaboration with the people working in the structure biology, right? So the keep talking about pull-down experiment. I'm wondering whether the pull-down experiment would help you eventually to, you know, identify the target of the compound.
Yep, that's a really good point. So to validate the target, so now on the computational analysis, we found some targets of the coniferous alcohol, right? So to confirm that, you know, this target is actual target in real setting, first of all, to save budget, I would suggest anyone who would like to do this to do the molecular dynamic simulation or MD simulation. So that would simulate the actual biological setting in computer, but the challenging thing about doing MD is that we actually require a very high performance computer, like very high specification, like Lanta in Thailand or those in A-Star in Singapore. Yeah, and it actually consumes a bit of the budget on investing on the high performance computer. And after that, after this MD simulation experiment results coming out and it looks promising, then we can do the experimental sort of validation later, because for experimental validation of the target actually costs a lot. So first of all, we need to have the purification of those targets, right? And then we need to mix them, well, simply speaking, mix them in the test tube and see the bindings, and then we can confirm with mass spectrometry, for example, to see the bindings, whether it's real or it's just simulating or imaginary. Yeah. So because of the limitation of time of my current MSc students, so we will try, you know, submitting the proposed target first to the lower than expectation journals, and then we'll see how it goes. But at the same time, we are now running the parallel study to do a experimental confirmation.